right, this is chapter 13. So in chapter 13, seed propagation. So this one, if you look at the word propagation, you're like, oh my goodness, it is really about making more plants through seeds. So your chapter outcomes or learning targets are describe seed morphology and development, understand the environmental conditions needed for optimal seed germination, compare strategies to break dormancy responses in different seeds, explain different seed propagation techniques, summarize categories used for selecting seeds, discuss production of high-quality seeds in seed saving and storage, and identify careers related to seed propagation operations and maintenance. So, seed morphology and development. We're talking about, so this picture shows you some zinnia flowers. They're talking about the seed itself as a reproductive unit. It is basically a baby plant. So when they say the word embryo, think of that's the baby plant. It's a result of fertilization. They do have a protective outer covering and storage tissue. So, and they can be dicots or monocots. If it's a dicot, they store tissue, basically the food source, in the cotyledons, and then in the monocots, it's in the endosperm. And you guys will be doing a lab with those seeds as well, so you can see the difference. This picture I love. It's kind of cool. You can actually take a tomato, slice it like this, and plant it in soil, and it's going to grow new tomato plants, because in here, if you've ever dealt with tomatoes before, there's a whole bunch of seeds. So this is histodifferentiation. That's a fancy word for the embryo and endosperm develop distinct characteristics and seed size increases rapidly. So the whole process is started through adding water, but it's talking about here. So cell expansion, seed undergoes a phase of swift cell enlargement due to accumulation of food reserves in the form of carbohydrates, fats, oils, and proteins. Maturation drying, the last stage, seeds have reached physiological maturity and rapid water loss. So you'll notice, like say if I have this tomato plant in my yard, as fall comes, the plant turns brown. If you leave tomatoes on it, they get brown and mushy and gross. If you let them dry, that's the seed you would save and actually use later on. Desiccation, that's drying of seeds, preparation for seed to be dormant. And then vivipary, that's unique natural phenomenon in which seeds germinate inside their fruit without maturation drying, which is the example here is going to be that vivipary, which is super cool. Apomixis, this is the production of seeds without going through the fertilization process. Some plants can do that where it's asexual. It's basically a genetic clone. So I know if I took the seed from this very plant, that flower I see should look just like this one. And that's actually a dahlia that you're looking at. So seed germination. Germination, this is one of the labs that you guys are going to do. Germination is just a way of saying that the plant's going to start growing. So this is just a seed. It looks like a wheat seed, where if you notice, it's added moisture, the seed's going to expand, and you have the plant starting to come out and then the root starting to come out. So the seed has to be viable in order for this to even happen. And it has to have good environmental conditions, and some plants actually have to have a special dormancy period that they have to go through before they will start to sprout. Seed germination is showing you the phases. So this phase, basically, they have imbibition, which is the physical process where water is rapidly taken up by the seed, hydrating the inner tissue. There's the lag phase, which is the period with little or no water uptake, but with many cellular activities that prepare the seed to grow. And then there's the radical protrusion, which sounds super cool. It's really just like right here, the image is showing you, it's that root starting to bust open through the seed coat and the plant starting to find its way. So growing point for the root is the radical, and the growing point for the developing shoot is the plumule. So it's showing like this would be the plumule starting to come up. And the cool part is the seed figures out like, oh, hey, sunlight's up here. I better extend that plumule up this way so the plant can grow. Germination rates. So when I buy seeds, it's going to say on the back of the packet what seed lot it's from, the germination percentage, how many of them actually germinated or started to grow, and the uniformity of it. And that's kind of, if I am doing a large crop of a certain plant, I'd want to know that so that when I go to harvest it, they're already at the same point. Environmental conditions for germination. Some plants need it to be warm. This is showing you, like right here, this is an image inside of a greenhouse where they have a heat mat underneath the plants to keep it warm. Temperature is usually the biggest factor. Temperature influences the seed dormancy control, the germination percentage, and the rate of growth. And then if you look too, it says cool temperatures need cool temperature seeds need cool soil temperatures. Cool temperature tolerant will germinate from 86 to 104 degrees Fahrenheit. Cool temperature requiring will not germinate if higher than 77. This image is primrose flowers, and they require cool temperatures and will not germinate at temperatures above 77 degrees. These are warm temperature seeds. They will not germinate if cooler than 50 to 60. So you have asparagus here, and then you have eggplant. And asparagus is kind of cool because it's a perennial, but you shouldn't pick it until like 2 to 3 years after you've planted it. You got to let it get established before you start ripping it out of the ground. This is talking about water. Water is very important for the fact that it causes that seed to start to, become larger. As it absorbs water, it triggers it to start to grow. And also it says seed priming. That is that whole process of hydrating and then drying out a seed for a greater germination rate and uniformity. And then it says plugs. Plugs are basically a fancy way of talking about this tray. Typically seeds are grown in these little plug trays, they call them, and then they'll take, as you can see, these are at that point where they should get transplanted somewhere else. They're getting too big for that plug tray. Environmental conditions for germination. Once again, you have oxygen. They have to have some sort of oxygen. If you just have them soaking in a glass of water, they're going to rot. They're not going to grow. They also have, there's some that are very light sensitive that require light. Most plants don't require light until it's that point where the plumule has come up and it can start the process of photosynthesis. Some species are actually inhibited by light, meaning that they won't grow until they're in darkness. And then you have some more, the environmental conditions. Light quality, obviously part of it is that you can do supplemental lighting. That's what this picture is trying to show you. I can force a plant to germinate in the middle of winter using heat, light, adding water to it. And it mentions this fun little word called photoperiod. We're talking about the length of daylight. Some of our plants that you see naturally occur in the wild, they are triggered, like our tulips come up in the early spring. They can handle the cold temperatures, plus they've gotten just enough light. And you'll typically see it happen as the day length is becoming longer. Light-sensitive plants, they have a dormancy that requires light in order to germinate. And after seedlings emerge, high levels of light are important for sturdy and vigorous plant growth. So seed dormancy. Primary dormancy occurs as the seed is shed from the plant. This picture is actually a lodgepole pine. We don't have those around our place. These ones require high temperatures of a fire to melt away the resin that covers the cone in order to release the seed. So they're a little dramatic. They need some fire in order for those seeds to get dispersed. If you look too, so dormancy, it allows the seed to germinate only when environmental conditions favor seedling survival. Quiescent seeds are seeds that have emerged from primary dormancy. And extreme stress may be due to drought, high temperatures, or low oxygen levels. Seed coat dormancy. So this is actually, in the picture here, is the Kentucky coffee tree seed pods. The seed coat can be as thick as 2 millimeters. So scarification is the first word on here, is the process of physically removing part of the seed coat to allow imbibition. Okay. So, imbibition is the rapid uptake of water by a seed during early germination. So, it's got a little spot on there, and most seeds have some sort of spot on them from their attachment to a pod or something else. Sometimes this can be mechanical, could be chemical, could be hot water. You can actually use sulfuric acid to wear away the coat of hard seeds. Kind of depends on what you are working with. Some of them, the mechanical ones, it could be scraping, it could be filing, cracking with a vise or a hammer. And obviously this one, the hot water one, you're going to do like 12 to 24 hours in hot water for some plants. Not all of them need these processes to happen, but you'll see some avid gardeners will soak their seeds. Chemical dormancy. So it inhibits germination. You can use phenols to do this, the naturally occurring chemical compound found in seeds. They also have abscisic acid. That's just a plant hormone, and both of them inhibit germination. So this one, they're showing you an image of what they call heirloom tomatoes. They're cut open to expose seeds covered with a layer of chemical that must be removed before they plant them. So they will do this. Heirloom varieties are typically ones that have been around for generations and they haven't changed them, so they're kind of unique. Morphological dormancy. Immature seeds that fall from a plant, they might need some temperature treatment of cold or warmth. Some examples that they have here in your pictures, there's the open poppy seed pod. Poppies have a red flower, and this would be after the petals have fallen off and the seed pod dries up and it bursts open. And this is a rhododendron with seeds. Physiological dormancy is controlled by embryo. It delays germination. You have to manually remove the seed covering, and then there's photodormancy, which requires light or dark to germinate. Stratification is a moist chilling treatment used to break dormancy in seeds of certain plant species. Seeds stored in moist aerated medium, specific chilling temperatures 1 to 5 months in seed beds can be used. Seed propagation techniques. You want to start with high quality seed selection of cultivars. You might be picking something specifically for the crop that you want to harvest. You have to look at the environmental conditions that you need to use for that seed too. They also talk about field seeding. The first thing you want to do is select your site, prepare that seed bed, select your high quality seed, time it correctly. Do any seed treatments that you might need to do. Make sure you have the planting depth correct. If I have a seed that needs to be 1/4 of an inch in the soil versus 1 inch, I could be basically planting it too deep and nothing will come up. Spacing. This is a big factor with your plants. If I don't have them spaced correctly, they're going to compete with each other and basically, kill each other. I need to have the appropriate equipment to do planting, and I need to water it. This is showing you like a field nursery seed bed. They're going to time their plants correctly. They also have these weird looking little bags, and these bags are actually little protective coverings for the plants as they come up. Greenhouse production, I could do this anytime I wanted to. I can force plants to do what I want through using a greenhouse. It's very controlled. It's ideal conditions for germination because I can control the temperature. It's protecting the plants from wind, from extreme weather. Consistent watering is very important, though. You don't want your seedlings to dry out. Seedlings are just, if you think of like babies, they're little baby plants. They need the, protection, and they're going to need you to nurture them and water them correctly. They also need, drainage as well. You can't soak them all the time. They have to have periods where they actually get to dry out the soil a little bit so they have proper aeration. Plug production. So these are going to be, if you can see here, these trays, they have little plants growing. It's kind of like we talked about before with the plugs. They'll grow in there for about 6 to 8 weeks, and that's at that point that greenhouses would sell them or transplant them. These are the phases of a seedling. So they're showing you this is radical emergence here. Cotyledon. Cotyledon is a fancy word for that first leaf that comes up. And then stage 3 or 4, it kind of depends on the plant, but it's going to start growing its true leaves after that. This is showing you some germination media. You want to have sterile stuff. You want to make sure it's fine texture. Soil to seed contact is important. Lots of moisture, good drainage, oxygen, and then adequate nutrients too. So this is part 1. We're going to continue on with a part 2 of this video for chapter 13, seed propagation. So check out for part 2.